Video is a short continuation on Unit 5 EOC review. I need to go over the cell cycle quickly. The cell cycle is just simply the cell's life cycle, and we've often depicted this using a circle. And the cell cycle is broken up into 4 major parts. And we'll just do equal parts even though they're not quite equal. There is G1, S, G2, and M. The G1 phase is the growth, primarily growth phase of the cell. This is when the cell is getting big. This is when the cell is making new organelles, making lots of proteins, that sort of thing. The S phase is when DNA is synthesized, and so DNA is replicated during the S phase. So growth, DNA replication, and G2 is preparations for division. And so M is actually cell division. Now M is divided into separate parts. There is, and the most of this is mitosis, and this is what we commonly associate with cell division, is the word mitosis. And mitosis is 4 phases. There is prophase, metaphase, anaphase, and telophase. Now, many will confuse this and put interphase in this as well. Now, interphase is the combination of G1S and G2. Those are all interphase, and inter is between phase. And so this is this phase between cell divisions. That's the way I think about it. And then, so after interphase, the cell enters into prophase. And in prophase, you have a cell where this is where all of the chromosomes condense. So whereas before you had a cell and on the inside the DNA was just kind of all over the place, in prophase the DNA actually condenses into these structures called chromosomes. And the chromosomes look like little X's because they are replicated. One piece has doubled itself. And a chromosome, just to look at a close-up of one, has a couple of parts that we need to be familiar with. That's from my sermon. You don't want that. All right. And so in the middle you have this little connector here. You have sister chromatids, and these are these pieces of the cell. Chromatids. And this is the centromere. It's like the connecting part there in the middle. And during prophase, the chromosomes condense down into this form that you see, and the nucleus, whereas there had been a nucleus in the middle of the cell that contained all of the DNA, like so. Now the nucleus is completely broken down. It's no longer there. And you also have these structures called centrioles that begin to form. And they begin to move towards the poles of the cell. So that during metaphase, that cell in prophase, the centrioles have gone to the poles of the cell, and the chromosomes have aligned themselves in the middle of the cell like this. All right, and the centrioles connect themselves to these centromeres. They're at the middle of the chromosome with these fibers called spindle fibers. And it's called metaphase because the chromosomes are in the middle of the cell. Next is anaphase. And anaphase, still have your centriole structures back here. These chromosomes, sister chromatids, are now separated, and the spindle fibers begin pulling them back towards opposite ends of the cell. And what you see is these cells begin to kind of pinch in like that, because what's getting ready to happen, the cell's getting ready to completely split in two. So that during telophase, or telophase, you'll sometimes hear it called, you have the nucleus, it's kind of beginning to form back. These chromosomes are kind of beginning to unwind as well, until the final, and then that's the last phase of mitosis, and the final act of the M phase is the division of the cells, which is called cytokinesis. Cytokinesis, which is the formal final division of the cells into two cells. Now, meiosis creates exact copies of the original cell. And so, let's say you have a skin cell here, and this skin cell is 2N, or diploid, because it has two copies of every chromosome, 2N, diploid. Here's your skin cell. Well, after mitosis, you're going to have two of those skin cells, and both of them are going to be 2N. This is different from meiosis. Meiosis only occurs with sex cells and is only done to produce gametes, whereas you start with a 2N cell, and that divides not once, but twice, to create 4 haploid cells, gametes. In this case, they're little sperm cells. Right? And so mitosis creates 2 diploid cells that are identical. Meiosis creates 4 haploid cells that are all genetically different from one another.